Ladies and gentlemen, thank you for waiting. Now we would like to start Lecture 1 by Professor Mario Capecchi. Let me introduce the moderator for this session, Dr. Osamu Chisaka, Associate Professor at Kyoto University.
Good
Dr.
morning,
Chisaka, please.
everybody. I am Osamu Chisaka from Kyoto University. I will be the moderator of this session. Now, I'd like to introduce the lecturer, Professor Capecchi, by way of a brief profile. Professor Capecchi was born in Verona, Italy in 1937. He received his Bachelor of Science degree in Chemistry and Physics from Antioch College in 1961, and his PhD degree in Biophysics from Harvard University in 1967. His thesis work, under the guidance of Dr. James D. Watson, included the analysis of the mechanisms of nonsense suppression, the initiation of protein synthesis, including the demonstration of 4-methionine tRNA as the initiator of protein synthesis, and mechanisms of protein termination. From 1967 to 69, Dr. Capecchi was a junior fellow of the Society of Fellows at Harvard University. In 1969, he became an assistant professor in the Department of Biochemistry, Harvard School of Medicine. He was promoted to associate professor in 1971. In 1973, he joined the faculty of the University of Utah as a professor of biology. Since 1988, Dr. Capecchi has been an investigator of the Howard Hughes Medical Institute. Since 1989, a professor of human genetics at the University of Utah School of Medicine. And since 1993, distinguished professor of human genetics and biology. He is also co-chairman of the Department of Human Genetics. In 2007, Professor Capecchi was awarded the Nobel Prize in Physiology or Medicine for his discoveries of principles for introducing specific gene modifications in mice by the use of embryonic stem cells. Today, Professor Capecchi will speak on the subject, gene targeting into the 21st century. Mouse models of human disease from cancer to neuropsychiatric disorders. Professor Capecchi, please.
Is my mic on? Can I be heard in the back? Yes? Good. What I'm going to do today is actually, I'm not going to go into any history. Instead, I will simply talk about science, and particularly projects that are ongoing in the laboratory, and then let the science speak for itself. Because that's essentially what scientists are comfortable with. I always tell my students the reason they're in science is because they cannot communicate with other people. If they were really good communicators, they would have become lawyers. They're comfortable in a lab, and that's good, and so that's why we do what we do. First of all, also welcome to all the students. You're the next generation of scientists, and we reach out to you, and we hope that you'll succeed in doing things way beyond what we're imagining today. I think that's one of the things that's nice about science, is simply that it always is changing. The problems we were working on 5 years ago are completely different from what we're working today, and they will be completely different from what we will be working 5 years from now. So, and I think what is important about science is to be able to be flexible and to take cognizance of that change and take advantage of it. That is, welcome it and then use it for your own particular interests. So I'm going to today tell you about two stories, simply to illustrate the kind of problems we're working on. The first will be modeling cancer, and then the second will be modeling a neuropsychiatric disorder. So we've decided to model sarcomas. And the first question you should ask yourself is why sarcomas. They're not as common as carcinomas, but there are reasons for working with sarcomas, and they're shown in the next slide. They first of all affect the critical population. That is, they affect children and young adults. Rather than being a problem of old age, it's actually, they're very aggressive, and they affect younger people. They're not studied as extensively as carcinomas simply because there are few numbers. They represent about 1% of total cancers. But if you're a pediatric oncologist, you see sarcomas. But they have also a number of advantages. I should point out that all of our models, all of our patients are mice. Okay, so if you're a modeler, you like to begin at the beginning. Do you know what initiates the cancer? And in terms of sarcomas, we believe we know the initiating event. There are roughly 25 different sarcomas, and most, I should also say, in biology, you can never generalize. There will always be exceptions, and keep that in mind. On the other hand, in terms of sarcomas, most, probably over 99% of them, are initiated by a specific chromosomal translocation. Two chromosomes break, and then they join together, but with opposite members. Everything is still okay, except at that junction, now you've created a new product, which is half of a gene coming from one chromosome and the other half coming from the other chromosome. And that is what initiates those particular cancers, and therefore we have, in a model, we can then generate that translocation product and allow us to initiate that sarcomas. Finally, I want to point out that they do have another advantage with respect to carcinomas. In carcinomas, genome instability is a hallmark. Okay? All of a sudden, many thousands of things are occurring in that cell, and you have to be able to pick out, filter out those that have a contribution to the cancer from the thousands upon thousands of events that have nothing to do with the cancer. In terms of sarcomas, often that translocation is the only thing that has happened. Okay, so progression, still you need multiple things happening. You have 5 or 6 events having to lead to progression of the cancer, but in sarcomas it's much easier to identify those secondary events to allow cancer progression. So those are the reasons we're working in sarcomas, and we've now modeled we've modeled 3, we're going on to a 4th one, and so on. And I think it's a paradigm that we can use over and over again. The other thing I will always be saying is we. And we is the royal we. I don't do the work, students do the work, postdocs and graduate students. And I'll try to give credit where credit is due. The first model I'm going to be talking about is synovial sarcoma. And when we started these experiments, this was what was known about synovial sarcoma, and that is they arise near joints. So here you see a joint, and here you can see the cancer, and this is the cancer when you've isolated from the human. Histologically, synovial sarcoma is complex. There are 3 histological subtypes. There's what's called monophasics, which are spindle cells, mesenchymal cells, biphasics, which include spindle cells as well as epithelial cells. And you can start seeing organization, essentially, to the tissue as if it's trying to build an organ. And then finally, common to many sarcomas, is what are called poorly differentiated, meaning small cells with large nuclei. So those are the histology. So that was known at the time. And in case of this, the translocation is between chromosome 18 and X chromosome. On chromosome 18 is the SYT gene, and it's also now referred to as SS18. And then on the X chromosome is SSX1. And what you can see is that, for example, in mice, there are 12 members of this family, and in humans, there are 9 members. And SSX1, SSX2 are commonly found in synovial sarcoma, whereas SSX4 is rarely found. There's one example of SSX4, and then all the other SSX genes are not found in the fusion gene. So these are both chromatin remodelers. SYT is always associated with activation domains, activation complexes. SSX is always associated with repressive complexes. This shows the fusion gene itself, and it contains almost the entire SYT gene and half of the SSX gene. It contains the activation domain from SYT and the repressive domain from SSX. So this new molecule is schizophrenic. It has both an activation domain as well as a repressive domain. So here we show our model. And depending on where the gene is expressed, we do use different models. In this particular case, SYT was controlling space, okay, telling you where this gene was going to be expressed. And it turns out to be expressed in all cells, so we didn't have to worry about space. Okay? And so what we have is we chose essentially the ROSA locus because it's a locus that's expressed ubiquitously through embryogenesis and even into adulthood. Okay? We didn't have to worry about space. So into that locus, we introduce a cDNA, which is isolated from a human cancer. And in this case, we is Malay Haldar, a very talented graduate student. And the first cDNA he isolated happened to have SSX2 as a member, so it's SYT SSX2. Okay? So we converted then into DNA. And then into that cassette, we also introduce EGFP and an iris, which is a ribosomal reentry site. So this cassette essentially then allows us to express the fusion gene as well as turn the cells green. However, between the promoter of the ROSA locus and this cassette, we put a stop sign and flank it with LOXP sites. And LOXP is the component of CRE, and LOXP makes a site-specific recombination such that this is CRE, the protein. It mediates recombination wherever it sees LOXP sites. So, and if you orient the LOXP sites in the same direction, in the presence of CRE, that stop sign is taken out, and then the expression of this cassette is turned on wherever we have CRE being made. Okay? So it allows us then to control where the cells, in which cells essentially the fusion gene is produced and also turns the cells green. Okay? So that's our model, and the reason we could do that is simply because we didn't have to worry about space. Okay. So the first thing we asked was simply what happens if you make the fusion gene in every cell of the body? And for that, we used HPRT because it's expressed in every cell of the body, starting at cell 1, going on to cell 2, and so on. So here's our mouse, has HPRT CRE, controlling where CRE is made. Then this is the other mouse, which had the cassette that I just showed you. Okay? So now we're producing essentially the fusion gene in every cell of the body. And what happens is shown in this slide. And what you get is a green blob. So that's good, it's green, that's expected. And this blob has all primary tissues. It has endoderm, ectoderm, and mesoderm, but it's completely disorganized. All the tissues are completely messed up, they're not making an embryo, and therefore the embryo is absorbed very quickly in a few days after birth. After inception. Okay? And you do not get any tumors. But that tells us the fusion gene is completely messing up development, okay? So having a strong effect on development. So where do we go next? What I told you is they occurs near joints. And the one thing that's in common about joints is that you want to be able to move them, and therefore it's associated with muscle. So we decided to look at muscle lineages. And we look at, make a CRE driver specific for each step of making muscle. So here it is, PAX3 and PAX7 CRE. These are muscle progenitor cells, and the end result is, again, embryonic lethality and no tumors. So then we decided to go all the way to the opposite end of the spectrum. That is, looking at fully differentiated muscle. And MIF6 is a marker of fully differentiated muscle, and so we made that CRE driver, again, meaning Malay Haldar. And here we got a surprise. So we cross MIF6 CRE to our mouse, and what we get is a classic myopathy. Here it shows the myopathy. So here's normal muscle, looking at it longitudinally, cross-section. And in the presence of the fusion gene, what you see is much smaller fibers, And then we also see wavy fibers and all the nuclei lined up. Now, when you make muscle, you have two stages. You have an early stage in embryogenesis where muscle is being generated from somites, and then that's when all the muscle is being patterned. Okay? Then in mice, about 2 days before birth, then the bulk of the muscle is made, and that uses satellite cells. Okay? And then those cells are also important. For example, if I exercise too much, I destroy muscle tissue, and satellite cells are used to regenerate that muscle. And a hallmark of satellite function is all the nuclei lined up. Okay? And that's shown right here. So what we're seeing is cells are dying and then are being replenished through the satellite system. And they continue to die, and then they replenish. And finally, after 6 to 8 months, the mouse dies of the myopathy, but it does not develop tumors. So that tells us that fully differentiated muscle is not a source of synovial sarcoma. So where do we go next? And that is myoblasts. Okay, these are cells that are committed to making muscle, but they're still individual cells, and they haven't fused to actually make a myotube. Okay? And we had a choice. We could either use MIF5-Cre or MyoD. These have overlapping functions. Okay? You knock out one, muscle's still okay. You knock out the other, muscle's still okay. You make a double mutant, no myoblasts, and no muscle. Okay? And Millet decided to make MIF5-Cre, cross it to the mouse. And what do we get? Our tumors in the right place. That is, they're always near joints. And I'll give you lots of data now to show you that there actually are synovial sarcoma. The tumors progress all the way through metastasis, and the pattern of metastasis in the mouse is identical to humans. For example, it's most commonly seen in lymph nodes, not surprising. Here it's showing you in the cerebellum. Why do we think that that is a metastasis? For two reasons. One is the tumors are much smaller, and two is MIF5 is never expressed in the cerebellum. Okay? We don't have muscle in our brain. Histologically, we see both monophasic and biphasic. In humans, if you have SSX1, at first presentation, it's already biphasic, whereas if it's SSX2, then it's mainly monophasic. We see the same thing in our mice. Ratio 10 to 1 is it's monophasic to biphasic. Okay? I should say that we gave this material blindly to a histologist that works in synovial sarcoma and human synovial sarcoma, and they could not distinguish the mouse material from the human material. Okay. Markers. There are over 25 markers specific for synovial sarcoma, and they're all found in this tumor. This is interesting. Myogenin. Myogenin is a marker of differentiated muscle, and it's not on. Okay. And in fact, all markers of muscle differentiation are not on in this tumor, and that's why it was not recognized as a muscle tumor. BCL2, 98% of human tumors express BCL2. We see strong expression of BCL2. Vimentin, a mesenchymal marker, strongly expressed in the tumor, and it's present in our mouse tumor. We've actually done microarray analysis to look at thousands of gene expression pattern, and it is very similar, indistinguishable from the human pattern. So that, by those criteria, we do have a good mouse model for human synovial sarcoma. This was sort of unexpected. So here's the tumor, and it's growing out of muscle. You see a little bit of green here, and that's autofluorescent. All muscle autofluoresces, but the tumor is green. That's expected because we're also turning on EGFP. But why isn't all the muscle also green? Remember that when you make muscle, you take a lot of myoblasts, fuse them all together, thousands of them, and then have a muscle fiber. So we expected it actually to be green, but the muscle wasn't green. Why is that? So we looked early in the mouse when muscles first being formed, and this is about 11 and a half days of gestation. So here you start seeing muscle formation, and what you can see is the green cells are there, MIF5 labeled cells that are producing the fusion gene, they're there, but they're not participating in making the muscle. Why aren't they participating? And the reason is they're dying massively. Okay, this is a tunnel assay, and you see enormous numbers of cells dying, except wherever there's cartilage. So here is a rib, and what you can see along the rib, all the cells are still there, they're viable and dividing. Okay? So that tells us that the rib is producing something that's maintaining those cells and then allowing them to divide and then acquire secondary event to allow then cancer progression. We're very interested in trying to figure out exactly what that substance is. The other thing that was surprising is that the mice are running around, okay? And yet we're killing so many of these myoblasts, except wherever there's a rib. Okay, so that indicated to us that I told you that MyoD and MIF5 have overlapping functions, but the overlap can be of two different types. You can have the same gene in the same cell doing the same thing, or alternatively, you have completely separate lineages. One lineage expressing MyoD, the other, the MIF5, we're killing the MIF5, and the MyoD is still there. We can test that simply by putting diphtheria toxin, again in the Rosa locus, put a stop sign flanked by LOXP sites, and now we cross it with MIF5 Cree and ask what happens when we kill all the MIF5 lineage. And that's shown in the next slide. And let's just look at this little part of the slide right here. Here's in the absence of Cree, here's in the presence of Cree. We're killing all those green cells, but if muscle wasn't there, essentially what you would see is a big concave because the muscle is there. Now, the shape of that area is completely indistinguishable, and therefore the muscle is still there. And what we're showing you here is simply what's happening to MyoD just at the time when you start killing these cells. And what you can see is the MyoD cells are taking off, whereas the MIF5 cells are now starting to die and then be completely dead if we looked out here in time. Okay? So that tells us that there are independent lineages, one expressing MIF5, the other MyoD, and we're killing the MIF5 and the other can compensate and the mouse is okay. Okay. So that's a summary of what we know about the biology. Now let's actually look molecularly to see what is happening, what is the fusion gene itself doing. Now, remember that SYT and SSX, they're associated, one with a repressive complex, the other with an activation complex, but they themselves do not have DNA binding domains. Okay? So they have to bind to something else that then tells them where to go on the DNA and have an activation or repressive effect. And the way you do that is simply make an antibody to that region where the fusion gene occurs. That's unique, okay? Now you have a completely new sequence, so you use that as an epitope to make an antibody, then you can use that antibody to bring all the proteins down, and then you can identify all the proteins that are associated with the fusion gene itself. And we know the identity of all of these proteins, and now the we here is Torsten Nielsen in collaboration. He's at the University of British Columbia. And I'll point out TLE1, which is a Drosophila protein called transducin-like enhancer of split, and then AFT2, which is activating transcription factor 2. And then obviously here this is the fusion gene itself. So those are components of that complex. This simply shows that the fusion gene is required to bring all of those proteins together. So this is a glycerol gradient, and simply big things sediment to the bottom, and smaller things stay near the top. And so in the presence of the fusion gene, then all of these components are together, forming a complex. In the absence of the fusion gene, then those proteins migrate separately. So it's necessary for making that complex. The other thing we can do is just make deletions along the protein itself. We just delete little pieces and then ask, where does ATF2 bind? Where does TLE bind? And what we show is that the amino terminus of the fusion gene is required for ATF2 binding, whereas the carboxy terminus binds the TLE1. And TLE1 is a repressive, again associated with repressive complexes. This shows that we can disrupt the complex by just making RNA, interfering RNAs, to the fusion gene. So in the absence of that interfering RNA, again, these proteins sediment at the bottom. In the presence of interfering RNA, then we disrupt the complex, and the proteins sediment separately. So it's necessary and sufficient. So here is the actual complex, and this is made from many of these different studies. So here's ATF2. ATF2, the activation, it's a way of sensing the metabolism of a cell. It's actually looking at cyclic AMP binding. It binds to cyclic AMP responsive elements. Okay, and wherever there is, you want to turn on genes in the genome, then that protein binds to it and activates those genes. Then it binds the fusion gene itself, which in turn then brings TLE1 to it, and then it brings essentially polycomb group complexes, again represses complex, as well as histone deacetylases. And then as a consequence, all of these target genes, ATFT genes, are now turned off. And then that then is responsible for initiating this cancer. Okay? So it's turning essentially an activation complex into now a repressive complex. And one interesting, for example, there are many target genes have been identified, ATF2, EGR1, early growth factor 1 is essentially a tumor suppressor gene itself. Okay? So you're turning off essentially that tumor suppressor and then allowing essentially now the cancer to progress. Okay, so what happens if we disrupt that complex, again using interfering RNAs? So here's simply looking at growth of the cells, a tumor cell. And we're looking both, these are human synovial sarcomas, and these are mouse lines that we generated in the lab. And what you can see is the growth rate goes down if you interfere with any aspect of that complex. Okay? Not only does the growth rate go down, but the plating efficiency goes down. And then finally, apoptosis is induced specifically when you interfere with that complex. So that says that complex is important and requires for maintenance of the tumor in cell culture. Okay? Here's simply a heat map looking at 10 different direct targets of ATF2, and then also looking at the heat with respect to TLE1, SS18, as well as the fusion gene. Okay. So, and this is two related sarcomas, and what you can see is the heat map, green meaning low activity, red meaning high activity, is very different in other sarcomas. And if we actually isolate the complex on the DNA and then sequence that complex, this is by chip sequencing, what we can see is the complex is sitting right on top of the cyclic AMP responsive elements. Okay, so that's right here, and this happens to be for the gene EGR1, the tumor suppressor. And we've done that for all the different target genes, and again, the complex sits right at that site, turning off those genes. And here's what happens essentially if you now interfere with that complex. You look at the expression pattern of those target genes, and they go up relative to control. Okay? Now we can also, since histone deacetylases are involved in repression, we can use drugs essentially that interfere with histone deacetylases and ask, do they disrupt the complex? And that's shown here. Romidepsin is a common histone deacetylase inhibitor and a drug made against those inhibitors. And what you can see is the presence of that drug, we disrupt the complex. Not only do we disrupt the complex, but and here's how we disrupt it. Essentially, it interferes with this junction right here. So now TLE is no longer able to bind to the fusion gene, releases the repressive complex, and those promoters now turn back on again. And that's shown here. So here we're looking essentially using two different drugs against histone deacetylases, and we can see that in the presence of the drugs, now we turn those particular target genes on, both in human cells as well as in mouse cells. Okay. So that has now resulted, since histone deacetylases have already been used essentially for human cancers, for example, small lung cancer, then we can ask now, do they work in humans? And so now there are clinical trials going on, 2 independent ones, asking, do the acetylases work in human synovial sarcoma? The other thing I want to point out is, remember I showed you that BCL2 is very highly expressed in these cells. And there are lots of BCL2 inhibitors have been made. Okay? A common class is shown in the next slide. This is navitoclax. And it's made against essentially the BH3 domain of BCL2. And it's a mimetic, and so it interferes essentially with interactions of BCL2 with other proteins, for example. And we can ask what happens. And in humans, these inhibitors, again, work, but then what happens is that other BCL2 members that do not have a BH3 domain are now then turned on, and therefore then essentially tumors escape from BCL2 function. And the escape route is shown here, MCL1 and BCL2A1. They don't have BH3 domains. They're turned on, and then the tumor becomes resistant. The human tumor becomes resistant to these drugs. But what we noted was, in terms of synovial sarcoma, those particular 2 escape routes are actually specifically turned off by that same mechanism that I just showed you. Okay? And for example, here we're seeing MCL1, and what we can see is in synovial sarcoma, they're expressed very lowly compared to normal. Okay? So it turns out that those particular escape route is turned off in synovial sarcoma. This happens just by serendipity. But what it told us is that these drugs may be very sensitive to synovial sarcoma. So we could test that, and that turns out to be the case. So here we're simply showing you drug concentration using a BCL2 inhibitor, and it's very sensitive essentially to concentration of that inhibitor. More interestingly, doxorubicin is the most commonly used drug in humans, in children, against essentially synovial sarcoma. And what you see here is simply a synergy between doxorubicin and the BCL2 inhibitor itself. Again, this is in cell culture using either human cell lines or mouse cell lines developed in the lab. We can also do it in vivo. We can add the drug to the mouse, and then what we can see here is simply the mass of tumors drops very rapidly. This is after 2 weeks treatment in a mouse having synovial sarcoma, and we can see the mass of tumors goes down, as well as the number of tumors rapidly goes down. And as a consequence, now we're just setting up clinical trials to test BCL2 inhibitors with synovial sarcoma. Okay. So now I'll turn to the second part of the talk, and that is modeling neuropsychiatric disorders in the mouse. And you might think that mouse is a bad system for looking at human behavior, and I hope to convince you otherwise. Now, we got into this by serendipity. We've been for a long time studying Hox genes, and Hox genes are important for establishing where things are made in your body, to make sure your head is at this position, your arms are here, all the organs are in the right place, and so on, so that you're a functional human being. And the nice thing is that the genes, the order of genes on the chromosome, tells you where these genes are operating. For genes at this end are operating at the anterior part of your body, here in the middle part of your body, and all those genes there are working at the bottom of your body. Okay? And here we're comparing essentially Hox genes in Drosophila and in humans and mouse. The exact same patterns present in humans and mouse. And what you can see is there's been an enormous expansion of these genes. There's been a quadruplication, as well as ABD members being expanded. Okay? Now, in this case, we, as Joy Greer, another graduate student, we've knocked out all of these genes, and we know what they're doing. Okay? And her project was HOXB8. And she knocked it out, and she's looking at the mouse physically, and she couldn't see anything really physically wrong with it. Okay? If we inactivate, even make a heterozygous for any of these other genes, we still see a phenotype. And yet with HOXB8, we couldn't see anything. So she did, well, if I can't see anything physically, maybe I should look at behavior. And what she did was simply take an infrared camera and take movies for 24 hours and ask what is the mouse doing at different time periods. Okay? The only behavior that she saw was altered was grooming, and that's shown here. All animals groom. Okay? You go into the shower, you lather up your hands, you start working on your head, and you work down your body. And that's what this mouse is doing. Essentially, it wets its paws, starts working on its head, works on the rest of the body, and ends up with the tail. This is what's called syntax. It's very reproducible. Over and over again, they do the same thing. The syntax is not affected in these mice. The syntax is identical. What's different is simply how many events and how long, how much time they're spending with each event. So it's the duration and a number of incidents of grooming versus non-grooming. Okay. And here's an example of her data. So these are SIB pairs. These are males and females. They're matched for sex. So they're 2 males or 2 females. The gray bar is a normal mouse SIB pair, and then the mutant is the black bar. And what you can see, for example, with respect to eating, they're spending the same amount of time eating. And she looked at many, many behaviors: running around the cage, building nests, running upside down the cage, drinking, eating, you name it. The only thing that was different is amount of time spent grooming. And that's shown here. And what we're seeing is that the mutant mouse, spends twice as much time grooming as normal. Okay? It's also independent of genetic background. This is an actual cross to a wild-type mouse that is from a field, okay? But we're following genotype. And we can also induce grooming. We simply spray water on a mouse, and that makes it start to groom. Okay? And we can measure then how long is it spent grooming. And again, you can see the mutant mice, if they have HOXB8 homozygotes, then they're spending twice as much time grooming as a normal mouse in a completely wild-type background. So it's independent of genetic background. I told you one slight error. We're taking movies over at 20. Disconnected. I can just hold it. Is this working? Oh, I see. Now, can I be heard? Okay, in the back? Good. So I told you one inaccuracy. If you're taking behavior over a 24-hour period, if you do too much of one thing, you've got to take it away from something else. And what they take it away from is sleeping. So they're spending less time sleeping. Okay? Relative controls. I'll show you a film to show you that it is pathological. Uh-oh. Whoa. Good. So what you'll see is when the mouse moves to the side, that it continues to groom one area, and then you'll start seeing removal of the body hair. So the mouse it keeps grooming, and then it'll turn, and there it's removed all the body hairs. So it continues to groom until it removes its body hair. This is very similar to OCD, a spectrum disorder called trichotillomania, where humans essentially compulsively remove their body hair. Okay? It's pathological, because they continue to groom beyond that until they have actually lacerations. And at this point, we put the mouse to sleep. And people with OCD, for example, often are wash their hands until they actually have lacerations on their hands. Okay? So it's a very strong compulsive reaction. This is also sort of unexpected. This is a normal mouse put in the same cage as a mutant mouse, and it grooms it. But the pattern is completely opposite. Before, it was removing all the body hair on the ventral side, the tummy side, whereas now it's removing what's exposed to it on the other mouse, a wild-type mouse in the same cage, on the back side. Okay? Now, this is important because it tells us that it can't be something to do with, for example, itch. It's not a peripheral nervous defect because it cannot feel, essentially, the itch on another mouse. So that indicated that it's somehow involved the central nervous system and the brain. And that's the source of this behavior. If that's true, this gene should be expressed in the brain of an adult mouse in the forebrain and midbrain. And the reason this is surprising is that Hox genes are normally not involved in patterning the midbrain and the forebrain. Hox genes stopped essentially when you were a crocodile, when you were mainly hindbrain. Okay? And it's very heavily involved, Hox genes are heavily involved in patterning the hindbrain, but have nothing to do with the midbrain and the forebrain. And here we're looking at the forebrain. And the mutation that Joy generated was these are very simple genes. They're just 2 exons. So she put a nonsense codon in one and a LOXP site in the other, and it makes the message bigger, and therefore we can see the mutant message versus the normal message. Okay, so here's a heterozygous. You can see both the normal band and the mutant band, and here's a homozygous having only the mutant band. This also tells us that it's not a neurodegenerative disease because the strength of these signals are about the same. So the cells are there, but now HOXB8 is defective and leading to pathological behavior. Okay? Now, at this point, we switch to a different we, and this is Shakwon Chen. Okay? And taking movies is extremely robust, but it's also extremely time-consuming, labor-intensive, because you're looking frame by frame, what is the mouse doing, and jotting it all down, 24 hours of film, and then doing that analysis. So now what we have is very simple platforms that allow us to automatically measure that activity. What they are is extremely sensitive to vibrations. And no matter what you're doing, you give out a different vibration pattern. If you're drinking, you have one vibration pattern. If you're walking around, it even keeps track, essentially, where you are on the platform. If you're eating, different vibration patterns, sleeping, grooming, scratching, you name it, it gives out a different vibration pattern. We have an algorithm that interprets that vibration and does it all automatically for as long as we want. This is over 40 different mice now, and what we can see again is the mutant spending twice as much time as a wild type in terms of grooming. So this is a very easy assay and allows us to quantitate it. Now, I told you that this gene is expressed in the forebrain of the mouse, okay? But the expression pattern in the adult is quite low, and it was all salt and pepper. It was difficult to identify which cells in the brain were expressing HOXB8. Okay? So what we do is simply lineage analysis. We take the Rosa locus again, put GFP in it, put a stop sign flanked by LOXP site, and now crosses with HOXB8 Cree. So wherever HOXB8 is expressed, those cells will permanently turn green, and we can see them. And here we got an enormous surprise. The only green cells in the brain are microglia. Here, morphologically, they look like microglia. If we use a pan-neuron marker for microglia, CD11B, it has coincidence of expression. It's not all microglia, a subpopulation of microglia, but they are microglia. So what I'm telling you, the immune cells of the brain, that's what the microglia are. They come from the blood, okay, and then go into the brain. And their rule is normally, if you have, for example, an infection by viruses or you have a stroke, they go there and clear up the mess. What we're saying is that HOXB8 microglia are controlling behavior, and that a mutation in that gene causes pathological behavior. Okay? When do these microglia get into the brain? There turns out to be 2 classes of microglia. There's one that gets in very early. These are actually primitive hemopoiesis in the yolk sac, and they get into the brain just as the brain is forming. Okay, and then there's a second class, which is labeled with HOXB8, that gets in after birth. And what we can see here, P2 means 2 days after birth. Here's the choroid plexus. Here's the ventricular surface. We start seeing buildup of green cells at that point. Remember, we're crossing it to a GFP reporter gene, so we can follow those cells. And then by 2 weeks, they're spread out all over the brain. Okay? So that tells us that they're behaving differently from the majority of cells. And these represent about 40% of all the microglia. If this is true, then blood should express our gene. And here we're simply taking blood, spreading it according to whether markers for myeloid cells, B cells, T cells, in the absence of Cre and in the presence of Cre. And we can see that whole population shifting. So it's expressed both in lymphoid and myeloid cells, and so that tells us it's either stem cells, hemopoietic stem cells, or progenitor cells capable of giving rise to both myeloid and lymphoid cells. Consistent with it being stem cells, here are two common markers of stem cells, c-KIT, SCA1, and these are the double positive in the absence of Cre and the presence of Cre, and again, that whole population shifts over, saying that the stem cell population is labeled with HOXB8. Is there a phenotype with respect to the microglia themselves? Now, the Cre driver we used was an iris Cre, okay, and we purposely used that because we didn't want to destroy the allele itself, the gene itself. But that has a problem. We now can't follow the mutant cells because one gene will always be wild type and the other will be mutant, so that doesn't allow us to follow the cells. But what we can do is simply count total microglia. And what we could repeatedly see is about a 15% reduction in total microglia, and that's very reproducible. However, this may be an underestimate if the other population compensates. It knows that there's fewer of these cells, and therefore it's making more of the other. So 15% is an underestimate, okay? So it's 15% or more, or it could be just 15%. We can't distinguish it because we can't follow the mutant cells. But it makes a very strong prediction, and that is we should be able to cure the mouse with a bone marrow transplant. Okay? So first of all, can we get microglia into the adult mouse after birth? And so we use bone marrow that's GFP labeled, and sure enough, here's 4 weeks after bone marrow transplant. Again, we start seeing buildup at the choroid plexus, at the ventricular surface, and then by 3 months after transfer, now we see bone marrow, green cells all over the brain. And our transplantation efficiency is quite good. It's about 75%. Okay? But remember, what we're doing is radiating the mouse to destroy its own bone marrow, then replenishing it now with new bone marrow. So now we can do the real experiment. We can go to the mutant, radiate it, put in wild-type bone marrow, and ask what happens. And that's shown here. So here is 3 weeks after transplantation, starts removing its body hair. If I showed you experiments 2 weeks later, enormous numbers of lacerations. And what we can see with the transplantation 3 months later, now all the lacerations are healed, all the hair is back, and this mouse is permanently cured of that behavioral deficit. And here we're looking at time spent grooming. Here's wild type, here's the mutant, and here's the rescued mutant, and it's completely gone back to normal. Okay, so with a bone marrow transplant, we can cure the mouse for life. We can do the opposite. We can put mutant bone marrow in a normal mouse and transfer essentially the phenotype. Okay, so that tells us it's both necessary and sufficient. Now there's a problem. Hox genes are pleiotropic. They do hundreds of things, thousands of things probably. There are 1,000 different target genes for every Hox gene. And it turns out that HOXB8 also has deficiency in sensitivity to pain and noxious agents. Okay? So we had to follow that up, and not only that, but actually another lab published that our results could be explained simply by these nociceptive defects. Okay, so let me first of all show you the nociceptive defects. So here's a cross-section of the spinal cord, and this is a pan-neuronal marker, and if you counted nuclei here, what you would see in the mutant, there are fewer cells there, and they're more disorganized. That's much more obvious if you actually look at markers. So here we're looking both at markers of the sensory neurons as well as the interneurons which receive the connections from these sensory neurons. Okay, so these are the sensory neuron markers, and we can see here's the wild type pattern, here's the mutant pattern, and so you can see that there are fewer neurons in the sensory system, there are fewer interneurons receiving it, and they're more disorganized. You'd have nice lamellar structures here, and they're sort of more spread out. So that's the phenotype with respect to the spinal cord. And you can also behaviorally test this simply by putting a mouse on a hot plate and then asking how quickly does it react, how quickly does it jump. And that's shown here. So here's a wild-type mouse, here's a mutant mouse, and it takes it longer to react to that, to heat. Okay, and the same thing would be true to noxious agents, but this is a lot easier to quantitate. So that's the phenotype. Now, the first indication that we could actually separate those two phenotypes, okay, one is noxious agents, the other over-grooming and pathological grooming, is the rescued experiments themselves. So here is a rescued experiment. So here's the wild type, here's the mutant. How quickly do they react to this hot plate? And what you can see is the rescues is not very much affected by completely restoring it in terms of the pathological grooming. Okay? So it still is insensitive to pain. Because that tells us that possibly we can separate those two phenotypes. Okay? And the way to do that is simply conditionally. We inactivate it in the spinal cord, ask what happens, inactivate it in the bone marrow, and ask what happens, and see what the consequences are. Okay, so for inactivating in the bone marrow, we use Ty2-Cre. Ty2-Cre is expressed in hemopoietic stem cells, but also endothelial cells. Okay? So it has that shortcoming. Okay. And here's the actual experiment. First of all, does Ty2-Cre label microglia? And the answer is yes. So they turn green, so that's good. And here's the actual experiment. And what you can see is by just inactivating it in the bone marrow or endothelial cells, it starts to remove its body hair. It spends more time grooming, but it does not affect insensitivity to pain. Okay? So we've separated in that direction. We inactivate in one in the bone marrow, we get all the pathological grooming defects, but do not affect the hot plate assay. We can also look at the spinal cord itself. That's shown here, and it shows the pattern of wild type. So here is the wild type pattern, here's the mutant, here is the inactivation in the bone marrow, and it's similar to the wild type pattern rather than the mutant pattern. Okay? We can do the opposite, inactivate it in the spinal cord, and for that we used a paralog of HOXB8, C8, has the exact same expression pattern in the spinal cord, shown here, but it is expressed a little bit in the microglia. Here's a spinal cord pattern. If I showed you HOXB8, it would be indistinguishable. We see a few green cells, but it's 15-fold reduced compared to HOXB8. So here's the number in HOXB8, and here's using HOXC8. Okay? So there's a few cells, but very few green cells in the brain. And here's the actual experiment. It has no effect on removal of body hair, no effect on time-space grooming, but completely shows the insensitivity to pain. So that shows that we've gone in the other direction and been able to separate essentially out those two phenotypes. Here's actually looking at the spinal cord, and again, here's the wild-type pattern, here's the mutant pattern, and now just inactivating in the spinal cord gives back the spinal cord defects. So in summary, what I've showed you is that microglia are controlling behavior. And the question is, how is that possible? How do microglia control the output of a circuit, a neural circuit? And there are multiple ways that can happen. So here's microglia. For example, they produce cytokines, they produce chemokines, they produce even glutamate. Okay? So they could be producing substances that then affect the neural circuit. This is the OCD circuit. This is, for example, simply you take people and put them in a functional MRI machine. If they have OCD or trichotillomania, these regions of the brain are overly active. If they react to drugs that suppress that activity, then that activity goes down, and that's defined essentially the striatum, dorsal aspect of the striatum, the neocortex, the motor cortex, the thalamus, and that circuit is the same circuit that's actually used in learning repetitive behavior and then also modulating repetitive behavior once you've learned that circuit. Okay, so we know where to look to see whether there's a defect, and the question is how are these microglia interacting with the cells in this circuit to then modulate its activity. There's another more interesting possibility. Waki et al. Actually here in Japan has shown the microglia, resting microglia, have these enormous protrusions, sotopodia, okay, and they're waving around in space, and they're always moving. always sensing it. And you can actually calculate that in about an hour, they can cover the entire parenchymal space. Okay, so in a sense, they can sense the entire space in about an hour's time. Okay, and further, what Waki showed was that then all of a sudden these pseudopodia stop, and what do they do when they stop? They're actually surrounding a synapse itself. And they completely surround it, and the membranes, essentially, if you look at the EM, you see that membrane completely around the synapse. And the question, why are they doing that? And can they actually modulate that activity across that synapse and somehow affect it? And now, through the studies of optogenetics, you have the perfect tools to start to address these questions. We can activate it in microglia, ask the neighboring neurons know about it. You can activate the neurons and ask the microglia know about it. That is, are they the same train of a set of events? Because with optogenetics, you can essentially give information by controlling pulse length. So here's another thing that was interesting. So here we're labeling now HOXB8 labeled microglia red, and then all other microglia, we don't have a specific marker that distinguishes that first population. But we can label all microglia green. Okay? And then all of this red are the inputs coming from the spinal cord. And remember, I showed you if you knock it out in the spinal cord, it has no effect on this behaviors. So we can neglect all of these, all of this red. But if we look individually in any particular region, what we see is areas, and when we look at that ratio, we see areas that are high and areas that are low. And it turns out where it's high is that same circuit that I simply showed you just a while ago. That is, that same OCD circuit is where HOXB8 microglia are high and the other resident microglia are low. So that also tells us that these cells, these microglia, are not uniformly spread out throughout the brain, but they have some way of knowing where they should be, and the question is, how. And so we can start to address those kind of questions. Okay. So in summary, I want to stress a couple of things. Okay. One is a connection essentially between immunology and behavior. It's been known in many, for 30 years. If you look at monopolar depression, bipolar depression, autism, schizophrenia, even Alzheimer and OCD spectrum disorders, there's long been known association between the immune system and behavior. Okay, these patients often have defects in the immune system. The question is, what's the relationship? Are you depressed, and therefore your immune system isn't working well, or is your immune system not working well, and therefore the risk of becoming depressed is higher? Okay? Cause and effect weren't clear. Here we're showing you a direct cause and effect relationship. Defective microglia give rise to defective behavior. Okay? So that, because we know a lot more about our immune system than we know about how our brain works, what that tells us is when you have the defective deficiencies essentially in the immune system, does correction of that affect the behavioral output? Okay? So that's one possibility. The other question is, what's the relationship of what we're looking in the mouse relative to what's happening in humans? Okay, first of all, we're looking at HOXB8 mutant homozygotes. Okay? Fortunately, 3% of people have OCD. 3% of people have trichotillomania. All populations have been looked at all throughout the world. It's the same level throughout the world. Okay? It's very common. However, 3% of people aren't homozygous for everything. There are populations where you do have a lot of homozygosity, but fortunately, it's not worldwide. Okay? So that tells us, in essence, that a HOXB8 mutant homozygote is not likely to be causative for trichotillomania in humans. Okay? However, what we're saying is that there may be a relationship. First of all, 3%, if somebody is 3%, that tells us either there are a lot of genes that give out the same output, or alternatively, there's a common polymorphism out there. Turns out that there is a common polymorphism in HOXB8, present in about 20% of all people, and it does track essentially with the disease. Okay? So that shows that what we're looking at may not simply be something in mice, but also common to people. But in people, what you would have is a HOXB8 mutation, for example, or a polymorphism, and then something else. And what is a something else? It would be defects in the immune system. Okay? So we know where to look, essentially, for other causative events, which together would give you rise to the same effect of it being a HOXB8 mutant. Okay? The other thing that I should point out is that a lot of bone marrow transplants have been done, over 400,000, and this is mostly for cancer. Okay? And I'm telling you, these diseases arise at the 1, 2, 3% level. So there's a lot of overlap in that data set. So I've gone to many different bone marrow transplant centers, asked them, have they seen anything funny with respect to behavior? And they don't keep that data. Okay? So the experiments have been done, but I do have a pile of emails about that high from patients. They say remarkable things have happened in both directions. Okay? So I think it's something we should keep in mind in terms of when you're getting a bone marrow transplant, you may be getting much more than you think you're getting. Okay? So that I'll stop there and open it up for questions, and thank you for your patience.
Thank you very much, Professor CAPECCHI, for your wonderful lecture.
Thank you.
So now the floor is open for discussion.
Thank you.
Are there any questions? Okay, the front row.
Thank you very much. My name is Mohammed Al-Tahlawi from Egypt. I want to ask you, could we explain the grooming that occur in mice on immunological basis rather than neuropsychiatric basis, means gene mutation causing some sort of allergic reaction due to antibody-antigen reaction that in turn causes severe itching desire that causes this grooming maneuver, evidenced by the changes in T and B cells and the improvement of grooming after bone marrow transplantation. This may be just allergic action rather than neuropsychiatric changes.
So the question is whether what we're looking at is some kind of allergic reaction between the bone marrow. I mean, that we can do it both directions tells us there's something different about that bone marrow, whether it comes from a HOXB8 mutant or a wild-type mouse, okay? Because we can both cure it by putting good bone marrow into a mutant, or vice versa by putting mutant into a... So if there's an allergic reaction, you would think that that would be independent, essentially, of that particular experiment. The other thing that's I should point out is that we don't think that this whole system is set up for looking at trichotillomania. Okay, it doesn't make sense to say you've set up this elaborate system, particularly to control that behavior. What are we really looking at? Okay, and I think what we're really looking at is anxiety. If you ask any psychiatrist, I mean, and I've talked to hundreds of people with OCD, trichotillomania. They tell you if they're anxious, they're in their pathological behavior, and when they're relaxed, then they're not in it. So there's a tight correlation, essentially, between anxiety levels and output, behavioral output. Our mice are they're off the scale in terms of anxiety. So, and there are many different anxiety tests you can do, and they are anxious mice. Okay? So I think that's what this system is actually set up to do, is to essentially control the level of anxiety, quiet things down, or when you need it, to be anxious. If a tiger's chasing you, you want to run like hell. And so then it's good to be anxious. But on other occasions where you want to communicate with each other, then you want your level of anxiety to drop down. And that's what this system is there to play, is to essentially alternate the level of anxiety. So I think, I don't think we're looking... I should also point out, when we're making a bone marrow transplant, you're right, we're also transferring B cells and T cells. But we can test that by simply using a knockout, essentially, a RAG1 or RAG2 knockout, which cannot make T cells and B cells, and ask what happens. And there it's interesting. The early phases of the pathology are not affected. But then the later phases, once you have lacerations, then you start requiring the T cells and B cells, and then you get inflammatory reactions. And it's exacerbated by T cells and B cells. But if you use bone marrow from a RAG1 mutant, then the early part all takes place. So I think that says also that that part of the immune system probably isn't critical. Yes?
Okay.
Hi, my question relates to your sarcoma work. Bruce Spiegelman from Harvard has shown that progenitors for brown fat come from MIF5 positive cells also. Have you seen anything in brown fat regarding your sarcoma work?
Yeah. So the question is the MIF5 population affecting brown fat? There is a tight correlation, essentially, between muscle lineage and brown fat lineage. We haven't really looked at that aspect of the story.
Okay.
Hi. Thanks very much. My question is relating to over here. In the red.
Okay, good. You're right behind the light. Okay.
My question is relating to how these mice actually respond to an immune response that the microglia would normally respond to. Do they still respond normally, or is that also affected?
Sure. There, we have to make a new allele. We've actually made the new allele. That is, put essentially Cree into the coding sequence so that then we can follow mutant microglia and ask how has their activity changed. And that would be one interesting aspect of, simply asking, do mutant microglia, for example, respond to, you can destroy a little bit of tissue, and what would normally happen is all the microglia migrate to that destruction. And you can ask if now the HOXB8 ones do or do not. We can also ask whether they can wave around and make contacts with synapses. So all of that behavior now we can do by making a new allele, but we've just made it, and so we don't know the answer. But the other thing that microglia do, but I don't think it's associated with this, but it's important in terms of pathology in a sense, is that they're also, you make twice as many neurons as you normally need. I mean, that's simply a rule of thumb. And then what it does is simply weed out the ones that are weak and say we don't need those and destroy those, okay? And it turns out microglia are involved in that's what we call synaptic pruning. So that is one of the functions. But we don't think that's happening here because they would have to be very clever microglia. That is, remember, we're doing the bone marrow transplant in an adult, okay? So anything that's occurring during development, it can't correct, okay? And so it would have to know essentially in the adult which are bad connections and so on. And I think that it's possible, but I think that's a less likely good model than a direct modulation of the activity.
We can take the last short question. So, over there, yeah.
All right, thank you very much for the nice talk. I have a question related to the first part of your story. I'm quite surprised to see that EGR1 actually is quite related to the tumor formation of this certain kind of sarcoma, because we know EGR1 is importantly involved in the liver regeneration. So I'm wondering if you have seen any phenotype related to the liver dysfunction in your mouse.
Right. That we haven't looked, I mean, we haven't carefully looked at other behaviors. I should point out that, making translocations, the frequency is 10 to the -3. That's true in the soma as well as in the germline. In your lifetime, you have about 10 to the 17 cells, therefore 10 to the 14th chromosomal translocations. Some of these things do bad things, okay? In cancer, it's easy to see because that cell is amplified, and you can have lots of it, and it's easy to see that the translocation's there. In other tissues where you don't have that amplification, it was not possible until about 2 or 3 years ago with deep sequencing to be able to see, is there a translocation there? Turns out, for example, now I showed you that we got a myopathy with that particular translocation in the MIF5 lineage. Okay? Turns out now translocations are starting to show up in myopathies. So my guess is that there will be phenotypes associated with translocations. And some of them, for example, where do we have a lot of cells, okay, in our brain? So you should start looking for translocations because now the tools are there to be able to see it and see whether some of the neuropsychiatric disorders, for example, are associated with translocations. Because, it's enormous numbers, and if it occurs sort of early in the lineage, then it'll affect all the cells derived from that lineage.
Well, the time has come to conclude this session. Let's give a big hand of applause to Professor CAPECCHI. Thank you.
Thank you very much, Professor CAPECCHI and Dr. Chisaka. This will now bring an end to the morning program of the first day. And we'd like to make a few announcements from the Secretary along with the flow of the afternoon. As I mentioned this morning, I would like to repeat an announcement regarding photographing. Unauthorized photographing and recording are prohibited during the lectures and discussion sessions. Official photographers will be taking photographs of all the sessions, which will be shared on the website and published in the journals and other printed materials. Therefore, that will be issued by the organizers and their organizations. If you do not wish to appear on the publicized photos, please contact the Secretariat staff. Furthermore, please kindly refrain from uploading any of the unauthorized pictures and videos of this morning's lectures on public domains. Next, I would just like to make an announcement regarding the participant list. There are several changes made to the participant list passed out in your conference book. New lists have been printed out and will be distributed by the Secretariat staff after this. So to avoid any confusion, if you can please leave the old sheets contained in your conference book on the table and replace it with the new one. The staff will pass out the list right now. And as you're doing that, if you can just listen to what I'm saying through the microphone, lunch will be available in the group discussion rooms on the conference floor of the second floor of the Prince Sakura Tower. After this, please follow the signs and staff to the designated group discussion rooms. Upon arrival at the Prince Sakura Tower, please grab your lunchbox from room 6, which is at the end of the hallway. Our staff will direct you there as well. From there, proceed to your designated assigned rooms. After lunch, we will have group discussions from 1:40 p.m. to 3:10 p.m., followed by a coffee break in the adjoining room, Heian. However, for all participants, please return to this room initially after the group discussions and check where your seats will be for the flash talks on the poster abstract sessions. You'll be asked to take seats by your ID numbers, individual ID numbers. So until the actual flash talks start, please check where you'll be sitting and then enjoy some coffee in the next room, Heian. Following, please take all your belongings with you after this as you head over to the group discussion rooms. So grab all your materials, your conference bags, as well as your bags as well. And shall we proceed to the conference? Prince Sakura Tower. I will head in the front, so please follow me. Thank you.